I'm going to talk about some studies that my lab has been doing, and my lab is mostly focused on cancer research. My lab takes sort of a kind of a network view of the way that biology works, and it's sort of based on this kind of picture in that proteins typically don't act by themselves. They act in concert with other proteins. And for example, this group of proteins is involved in a certain function, and it's connected to this group of proteins, which is involved probably in a related function or a function that needs to be co-regulated with this function. Having this view of the cell gives a lot of input in the kind of experiments you do in the lab. So for example, with this protein, has been annotated for gene ontology. So there's some idea of what it does functionally inside the cell. The gray ones haven't been annotated functionally. So by knowing that this protein interacts with this protein, now you have a hypothesis that you can test. You can see if this protein is involved in the same function as this one, right? Typically, the idea is that in a normal situation, a non-pathological situation, the network is going to look one way. In a different situation, a pathological situation like cancer, this network is going to be reprogrammed. Okay, and so my lab is sort of interested in finding out how the network gets reprogrammed in different pathological conditions, and mostly we focus on cancer for a variety of reasons. Okay, the core technique in my lab is mass spectrometry. And basically what we typically do is some kind of affinity purification, typically an IP that we couple to mass spectrometry. And so we spend a lot of time optimizing things for getting the workflow to work really quickly. And so one of the things that my lab does that typically other labs don't do is we directly treat the immunoprecipitation with trypsin to create peptides, which then go in the mass spec. So typically people will elute these proteins off, maybe run a gel, maybe use some kind of filtration technique to purify the proteins away from the antibody bead and the antibodies that's associated with the bead. The reason that we can directly throw trypsin onto the antibody bead is the antibodies that coat the bead are typically resistant to trypsin. And they're typically resistant to trypsin because they have to live in places that are full of proteases. They have to live in your serum, they have to live in your digestive tract, they have to live in your tears. So the antibody is typically a really, really robust protein. It's very difficult to digest a native antibody with basically any protease that you throw at it, right? So you don't get a bunch of peptides coming from the antibodies that are coating the beads when you treat it with trypsin. And so in this way, we can take the mixture of peptides that come off the antibody beads and do liquid chromatography to separate them and then shoot them into the mass spec. And the mass spec is going to let us identify the peptides, and then from the sequence of the peptides, we can identify the proteins that were in the immunoprecipitation. So a typical about 1 hour of data collection looks like this, where each of these peaks are a different set of peptides or a different peptide that's being eluted. The mass spectrometer does a measure of these peptides. It tells you the mass of this peptide, and then it isolates this peptide and fragments the peptide. And so based on the fragmentation pattern, you can predict the sequence of the peptide. And so for me, this is the most important part of this workflow, because this is the part that is going to give me the data. Right? And a typical modern mass spectrometer will generate tens of thousands of these per hour. Right? So it's really, really a data-rich workflow. The problem is you're generating 10 or 20,000 of these spectrum per hour. Most of them are garbage. Most of them don't give a peptide match. Most of them don't match to anything. So one of the things that you end up with is you end up with a statistical-based workflow to identify the sequence that you're polluting with a bunch of data that is useless, right? So you end up making your search space really, really large with useless data. that screws up your statistics and actually hurts you in the long run for identifying peptides, identifying post-translational modifications, identifying proteins, identifying changes in your sample. Okay, so a lot of techniques have been used to try and clean up this data. And so typically the major one is you have some algorithm that takes a look at the whole spectrum and decides, is this spectrum likely to give a match, yes or no? If the answer is no, you throw the spectrum out and you never search it. Okay, so that is a spectrum by spectrum filtering technique. Because I'd spent a lot of time early on in my career actually annotating a lot of these by hand, I sort of understood the fragmentation pattern and the way the peptides behave inside the mass spectrometer. And so based on the way the peptide fragments inside the mass spectrometer, you can do a peak by peak filtering instead of a spectrum by spectrum filtering. And so, for example, now we use basically 3 kinds of filtering techniques based on the chemical ways that the peptide fragments inside the mass spectrometer. So the peptide should fragment between amino acids. Okay, so any peak pair that's separated by the mass of an amino acid we keep, right? Because if it's a peptide fragmentation, this should be the mass of a peptide. So this is Y6 and Y5, 1, 2, 3, 4, 5. So this is the mass difference here should be the same as a glutamic acid. Okay, and so we have 2 ways of capturing these what we call amino acid jumps. So that we have 2 filters based to pull out these. Okay, the other thing is if you fragment between 2 amino acids, this fragment and this fragment should weigh up to the whole mass of the peptide. Okay, so that's our third way of filtering is mass complementation. So any 2 fragments that weigh up to the mass of the parent peptide, we keep it. Okay, so these are really simple filters that I was able to write myself in MATLAB, and they run really fast because it's just simple arithmetic. It's subtraction and addition, and then you keep what you keep and you throw out everything else. Okay, so here is what is a positive predictive value of the spectrum. So in red is the raw spectrum before any filtering, and in green is the spectrum after the filtering. Okay, and so positive predictive value is basically a measure of the information content inside the spectrum. So basically you count how many peaks in your spectrum actually match the peptide that you're predicting it to be. So this is 2 different mass spectrometers, 2 different samples, but you can see that the positive predictive value of the raw data typically is around 10%, 5%, right? So this is pre-filtering. So 95 to 90% of your data is a waste. It's screwing up your statistic. It's making your life nuts. By doing the filtering, now it's up to 25, 30%. Right? So now you've really raised the true information content of your most important piece of data. Okay, so there's a lot of parameters that you can play with. Mass tolerances, how many peaks you end up keeping in the spectrum, blah, blah, blah. But in the end, doing the filtering based on the way the peptide is supposed to behave in the mass spectrometer gives you a really large plateau. So it's basically resistant to the different parameters that you might want to play with unless your filtering is really, really stringent and you're basically throwing away most of the peptides, right? So you can easily obtain 80% reduction in data. So your spectrum size is reduced by 80% and you're still keeping all your information content in the spectrum. Okay, so now you've really gotten rid of basically the garbage in your data, and you're keeping, by doing a peak by peak filtering, you're keeping the stuff that's important. Okay, why is this important? Okay, so if we take the raw spectrum and we do a search against the correct database and we compare it to the behavior of the same raw spectrum searched against the same sized database, but completely randomized. So these will be a mixture of correct matches and wrong matches, and these will be just the wrong matches, so no correct matches. And so if you compare these two lines, they're overlap for really, really a long time. So at this point, you cannot tell the difference between a spectrum that's annotated correctly and one that's annotated incorrectly. Right here, you're beginning to be able, because these two lines are beginning to separate, now you can begin to say that, yeah, these are right and these are incorrect. Okay, and so typically in the raw data, you need a lot of matches, 20 or so matches, in order to begin to separate this. By getting rid of 80% of the peaks that don't matter, you shift this all to the left. Okay, and so now the difference between real data and wrong data, it separates much earlier, and it separates much more steeply. And so what you typically gain by getting rid of the nonsense data or the data that doesn't help you in the search is this area here. Right, so really, really you improve the number of matches that you get, the quality of matches that you get, and the kind of things that you can find. Okay, and so we typically turn this crank for the mass spec on every project that I have going in my lab. Some point, every student, every postdoc, every technician is going to be shooting samples on the mass spec, because for me, the mass spec is really a fantastic tool of discovery. Okay, and so because we start with this sort of network view, in the end what we try and do is use a network view to help us interpret what comes out of the mass spec. And I'll show you a little bit about this with a project that we've had going in my lab for a long time on ubiquitin. And because ubiquitin is a small protein that's used to modify other proteins. And so this double glycine motif is attached to a lysine in the target protein, and ubiquitin itself can be targeted by other molecules of ubiquitin, forming chains of ubiquitin. Okay, and so my lab started out looking at chains of ubiquitin that are linked through lysine number 6 in the ubiquitin molecule. And that was because BRCA1, which is a very, very important tumor suppressor gene, is known to assemble K6 linked ubiquitin chains. And so what we wanted to know is what were the targets, so which proteins get K6 ubiquitin added to them, and what proteins actually bind to K6 ubiquitin chains. So we made a form of ubiquitin that can only form K6 chains, did a immunoprecipitation, digested everything off the beads, shot it on the mass spec, turned the data analysis crank, and we came up with some candidates that we started to study. And so the top candidate that we studied the most is this molecule called WIP. Okay, so we started off thinking that, okay, this is going to be fantastic. We're going to be looking at things that are involved in K6 linked chain biology, but that turned out to be a very naive view, because if we compare what comes down, if we do K6 ubiquitin IP or K63 linked ubiquitin IP, most of the stuff is the same. There's very few things that are specific to K6 or K63. And if we do a different ubiquitin chain, K48 or K11, the same picture is true. So you always end up with a bunch of things that are shared and very few things that are unique. And we understand why this happens. It was just because when we started this experiment, we were a little bit naive in the way that these ubiquitin chains work. Okay, but so from a typical IP, we can get a couple hundred things that we would consider to be potential hits, probably another couple hundred of things that we exclude, either because they're found in the control or because they're just common things that are found in a mass spec result, or they're just too difficult to work with. And so there's no point in even trying to study them, because you're just going to waste your time and drive yourself nuts. Okay, so this was our top potential hit. It wasn't known to be a ubiquitin binding protein or ubiquitinating protein, which are shown in red. And so we picked this one because we thought it would have a good chance of giving us some insight, new insight into ubiquitination. Okay, so it turns out that WIP will co-IP with basically any kind of ubiquitin chain you throw it at. It doesn't care. If it's K6, K11, 29, 48, 63, it doesn't matter. It will IP just fine. Okay, so we know now that this shows that WIP will actually bind non-covalently to ubiquitin, but we were also curious about whether or not WIP gets ubiquitinated itself, and it turns out that this is also true. If you look at the domain structure of WIP, there's a AAA plus ATPase and this Rad18-like zinc finger. At this point, there was no annotated ubiquitin binding domain, but by doing the classical thing of chopping this into bits and testing the different bits, we were able to map the ubiquitin binding domain to this zinc finger. All right, so for example, this is the zinc finger of, these are the zinc fingers binding to ubiquitin in vitro. Okay, and you can make a single point mutation which will abolish the ubiquitin binding in vitro and in vivo. Okay, we can skip this for now. Because we have this sort of instrument of discovery sitting in the lab, it's easy for us to sort of alter our IP strategy, and instead of looking for interacting partners, we can start looking for post-translational modifications. So we did this with WIP, and it's phosphorylated, it's acetylated, it's sumoylated, and of course it's ubiquitinated, and we can map the sites all along the length of ubiquitin. There's one ubiquitination site that's actually induced upon UV damage, and it sits right smack in the middle of this AAA ATPase domain, and it's a conserved residue that if you mutate it to any other residue, it doesn't matter what, the ATPase is dead, because this residue is absolutely 100% required for ATP binding. You change this lysine to anything else, ATP doesn't bind anymore. Okay, so it looks like this ubiquitination is functioning to block the action of WIP. Okay, and we can test that. So if we do an siRNA against WIP or WRNIP1, what ends up happening is if we use a luciferase-based assay to look for recovery from a damaged luciferase gene, knocking down WIP actually causes the recovery to happen faster. Right, so which this indicates that WIP is acting as an inhibitor of, typically acts as an inhibitor of DNA damage. And if we do a mutation in the ATPase domain, it kind of mimics this effect where you get a more rapid recovery of a damaged luciferase gene. Okay, so WIP is functioning as an inhibitor of DNA repair, and a functional ATPase domain is what is doing this inhibition. And so the UV damage is targeting the ATPase domain and inhibiting the ATPase domain and allowing the repair to go forward. Okay, since we know that at the time we started this was a unique ubiquitin binding domain, so it's very easy to bioinformatically go in and look for other proteins that have the same domain. And at the time that we did this, all of them were involved in DNA damage repair or DNA replication, except for one orphan one. And so we started studying this orphan one also. So if we look at the summary of the binding ability of all the different UBZ domains, they typically bind any kind of ubiquitin. They have no specificity. So there's not one of these domains that only binds to K6 or only binds to K48. It doesn't care. If there's a ubiquitin chain, it's going to bind to it. Okay. Both of these domains ubiquitin binding domains, you can make a similar point mutation and abolish the ubiquitin binding. The ubiquitin binding domain is required to recruit these proteins to DNA repair foci. So here is a WIP GFP construct that gets pulled into these foci. If you mutate the ubiquitin binding domain, it doesn't go there anymore. The same is true for the orphan one. The wild type one goes to the foci. If you mutate it, it doesn't go to the foci anymore. Okay. Both of them are regulated by UV damage. So if you stimulate the cells with ultraviolet, you get an increase in the ubiquitination and stability of WIP, but with UBZ1, basically it causes it to be degraded. Okay. I have a mass spec sitting in my lab, so I do an IP of UBZ1 to see what comes down, and I do an IP of WIP to see what comes down, and they're quite different. Right? So there's not much overlap between these two. If I take the ubiquitin binding domain from this one and the ubiquitin binding domain from this one without the rest of the protein and do the same experiment, then I get this picture. Right? Almost everything is the same, with very little bit of overlap. Okay, and so if you look at the proteins that come down and you do a typical kind of mapping experiment, interaction mapping experiment, it looks really different. Okay, so the take-home message here is that by using a mass spectrometry approach, we were able to identify a new ubiquitin binding domain using the kind of classical cell biology, molecular biology techniques that everybody uses. We can learn more about how this domain works. And the interesting thing about this domain, there's a couple interesting things. One is only found in proteins involved in replication and repair. And the UBZ domain has no real specificity by itself. In order for it to have specificity, it depends on the rest of the domains in the protein itself. Okay. More and more, proteomics is becoming quantitative. And so we've started doing most of our IPs in a quantitative manner. And we've also started doing some comparative proteomics, basically based on quantitative changes in the proteome. And so if you take a look at all the proteins that are in your sample, typically the mass spectrometer can only identify a subset of them. So some you will never identify. And even of the ones you identify, many of them you won't even be able to quantitate because there won't be enough protein there for you to get statistical confidence in the quantification. Right? So doing quantitative proteomics actually becomes a bit of a struggle. Okay, and so there's a bunch of factors that are involved in making quantitative proteomics even more difficult than normal proteomics. So typically quantitative proteomics these days, if you're doing an LC-MS-MS approach, is based on some kind of isotope labeling. Right? So you can either take your two samples, treat them differently for a long time, add a chemical reagent that adds the isotope, and then analyze them by a mass spectrometer, or you can label the cells in vivo with stable isotopes, mix them early, and then quantitate them by mass spectrometry. Okay, Baytul in my lab came up with a hybrid technique for quantifying proteomes. And so basically you take the cells, you label them in vivo, then you do you oxidize all the cysteines to cysteic acid. You can enrich these cysteic acid containing peptides using a mixed mode kind of chromatography. Then you do mass spec and data analysis. Okay, so this part is like the classical SILAC technique that you hear about all the time. And then this part is kind of the unique thing that she invented. And so the interesting thing about cysteic acid is it's very, very strongly negatively charged. And so you're adding a new negatively charged element to your peptide mixture, and you can use this as a means to fish out the peptides containing cysteic acid. Okay, so if we start with something like BSA and we turn the crank on this hybrid technique, we have some peptides that come out in the flow through. These are the things that don't stick very strongly to our mixed mode column. There's very few cysteine containing peptides in this mix. We wash, more of this non-specific stuff comes off, very few cysteines containing peptides come off, but when we do the elution to elute the cysteine containing peptides, boom, here they come, right? And there's not a lot of other peptides that are coming out that don't contain cysteine residue. These are typically things that contain multiple acidic residues, so they have like EEE or DDD in them. Okay, so this actually took a lot of time, and it works great with BSA, but we also needed to work on a real biological system. And what Baytool decided to use was to use a set of drugs and to study the changes in the proteome in response to treating cells either with sertanol or resveratrol. And these are both kind of related compounds because one is a suppressor of SIRT1 and the other is an activator of SIRT1. And SIRT1 is a sirtuin, and it basically functions as a deacetylase in the cell, and it's already been implicated in a variety of cancer pathways and neurodegeneration, et cetera, et cetera. So it's an interesting molecule in and of itself. Okay, so if we take a cancer cell line and we treat with sertanol, it basically induces senescence. So what we're doing here is we're staining for the beta-gal activity, which is the most common marker for senescence. Okay, so if we treat with a small amount of, relatively small amount of sertanol, we get about half the cells to become beta-gal positive. If we treat with even more, then we can get 90% of them to express this marker of senescence. The low dose one will stop dividing for a little while, and then we'll get some escape and they'll start growing again. But the large dose basically will arrest cell division for at least 10 days. We haven't looked past this. Okay, when we do the hybrid technique on these cells, one thing that we noticed is that we pick up a lot more acetylated peptides in the sertanol-treated sample, which is good for us because what we're trying to do, we're treating with a deacetylase inhibitor, so we're expecting actually more acetylated stuff to come out, which sort of verifies that the drug is working the way that it's more or less described. Okay, we can skip this. Okay, so if we look at if we compare sort of the classical method of doing comparative proteomics to the hybrid technique, you can see that there's a big jump in the number of peptides that you can identify and also the number of proteins that you can identify and quantitate. Okay, so we go from being able to identify like maybe 1,500 proteins to a bit more than 5,000 by doing the hybrid technique. Okay, not only do you get more proteins identified, but more post-translational modifications come out, more oxidized peptides come out, more phosphorylated peptides come out. I've already said more acetylated stuff comes out, more ubiquitinated stuff comes out. It just is a better workflow for discovery. Okay, so we turned the crank. We quantitated about 5,000 proteins in the sample. The distribution of the ratios between treated and untreated looks like this. We have about 140 of the 5,000 that are upregulated and maybe 37, 40 that are downregulated. Okay, this doesn't really help us do the next step, which is trying to understand if any of these things are actually relevant to the drug treatment. Right? So then you have to go into bioinformatics analysis. So you can use something like David, which will give you a functional, an enrichment of anything functional. So for example, we see enrichment in things that are involved in acetylation. This is perfect for us. Okay. You can skip this one. And so here is the list of the 38, 40 proteins that are downregulated. Right? This list by itself is pretty meaningless. Right? You can go through one by one and go through the literature and try and think about what might be happening. But we typically take a list like this and we map it on the interaction network. And so, for example, this list, which really looks like nothing, right? Actually, if you do the interaction network, what you find out is you have a really tight set of proteins that interact with each other that are all involved in the same function. And in this case, this is protein phosphatase 2. Right? So this is a set of proteins that are all downregulated that are involved in dephosphorylating stuff. Okay, so for me, the mass spectrometer is a really fantastic discovery tool if you're doing cancer research or microbial research or agriculture research. It doesn't really matter, right? You have an instrument that is going to give you a massive amount of data, and if you have a kind of even a semi-robust workflow for analyzing the data, really the biological information that comes out is really quite good and quite helpful in planning the real experiments that you do in your lab. Okay, so I'm done. This work was mostly done by Fabio, Martina, and Betul, who isn't pictured here, but she's sitting in the back there. Thank you.